Hello guys, let's begin with one more lectures today. That is about the glycolysis. Glycolysis is a very common pathway which is required for the energy production. Any organism which needs to produce, whether it's prokaryote or eukaryote, it has to pass through the glycolysis. There's a few exceptions to the glycolysis where the glycolysis does not happen. This is very. So most of the prokaryote and all the eukaryotes will have a process first, that is a glycolysis. Glycolysis means the breakdown of the glucose. So this is the first process which occurs into the cytoplasm and which is very important. Today our focus will be how exactly this is going to happen. At the same time, we'll be talking about how exactly we can regulate this. What is going to be the regulation mechanism, that also we need to talk about. Let's go to the basics of it. So there'll be a few things that we need to know when this process can be regulated. First, we have to check it level of energy. If the level of energy is low, so anabolism is unlikely to happen, or it is impossible, because you need energy in order to make some biochemical substances. Second one is the level of substrate. Third is the level of enzyme and their cofactor and their availability. For example, the lipoic acid, thymine, NAD plus, etc. And then the enzymes which are required, both quantity wise, repression or induction of the or expression of the information in the DNA of making of those enzymes. Second one is activity, whether the enzymes are active or inactive. So what kind of activation or inactivation is there, whether the enzymes are allosteric, so they may have the activator or inhibitor. So this is a kind of feedback control mechanism which is controlled by the final product. So once the final product is made, then it is going to inhibit the enzyme by bringing about some kind of covalent modifications. Fifth one is the compartmentalization. So all the enzymes, they remain in a particular compartment. For example, enzymes that we needed for the glycolysis to take place, it is found in the cytoplasm. And sixth level is the hormone control. Certain hormones, they're required to be triggered off in order to carry out the metabolic processes. And if you go into the definition, it is a organic molecule which is synthesized in the nano quantity and which regulates the many activities. We already have spoken about in the initial lectures how exactly they're going to play important roles. Let's not talk about it today. And then hormone regulation will affect the gene expression, for example, the steroids, and which or other, they can activate the second messenger. Basically, it could be the cyclic MP or the calcium ion, or it could be the inositol triphosphate. Let's see what happened about the metabolism process which is happening over here. So the glycogen, you can see the two arrows. One is if the glucose in excess, they'll be converted to the glycogen. If the glucose is lesser in the blood, so glycogen is going to be broken into the glucose. And from the glucose, there will be pentose pathways and other sugar can come to this point. And the further glucose is going to be broken down into the pyruvate, which have three fate. It could be converted to the lactate, called a lactate fermentation. Another one is it is going to be converted to the ethanol, that is called as alcohol fermentation. Lactate process occurs mainly in the muscles. And the third one is the acetyl coenzyme A, which will go into the TCA cycle into the mitochondria, where it will generate more ATP. Now we'll go just see a brief process of the glycolysis, where the glucose, look at the first step here, the ATP is required. So glucose is going to be converted to the glucose 6-phosphate, and the enzyme required for the process is the hexokinase. Now this glucose 6-phosphate will be converted into the fructose 6-phosphate by the enzyme phosphoglucoisomerase. And you can see this is the single step process which is going to be very important. Then the fructose 6-phosphate will be converted into the fructose 1,6 bisphosphate. You can see this is a reversible reaction. And then this fructose 1,6 bisphosphate will be broken into the dihydroxyacetone phosphate, we call DHAP, or glycerol 3 phosphate, with the help of the triose-phosphate isomerases. And the glyceraldehyde 3 phosphate further adds the NADH with the help of the glyceraldehyde 3 phosphate dehydrogenase is going to be converted into the 1,3-bis phosphoglycerate. Here the ATP formation will occur and will be converted to the 3-phosphoglycerate with the help of enzyme phosphoglyceromutase. That will convert this into the 2-phosphoglycerate, where the positioning of the phosphate group will change from the third carbon to the second carbon, and enolase will convert this phosphoglycerate into the phosphoenol pyruvate, and that will be converted to the pyruvate. Here again energy is going to be formed with the presence of the pyruvate kinase. Let's go into the detail of this pathway, because just knowing the steps will not solve the purpose. The purpose will be solved when we know the basics behind it. So first, what is the purpose of the glycolysis? Purpose is the metabolic pathway to convert glucose in order to get the energy. Here the one glucose molecule will give you the 2 pyruvate, and it will produce the 2 molecule of the each NADH and ATP. This is the net gain. All carbohydrate to be catabolized must enter into the glycolytic pathway, and the glycolysis in the center, as I told you in the beginning, both energy and the metabolic intermediate, because this is the process, and intermediate can be converted into the different form of biochemicals, including amino acids. Let's go to the phase 1, that is called as energy investment. We have seen that glucose is going to be phosphorylated. Let me tell you why does it happen. So glucose is relatively a very stable molecule, so it is very difficult to be broken to yield you the energy. So first the ATP is going to be expanded over here, and the one phosphate group is going to be attached to make the glucose energy rich compound. I mean the phosphate group is having negatively charged, so this is going to convert glucose into a very reactive molecule, or the one which is having the more energy, hence not a stable one. So we are compromising the stability of the glucose so that glucose can be further broken down into the other compound. So this is also called as priming. And here we require the 2 enzyme. One is a hexokinase, which is more than often used, and another one is a glucokinase, which can also be catalyzing the same process. Look at over here. This is your glucose, and the ATP will be hydrolyzed into the ADP, and the phosphate group will come and it can attach over here. So glucose is going to be converted to the glucose 6-phosphate, and here the energy is going to be delta G value is going to be minus 116.7 kilojoule per mole. So first energy is the investment, that is a highly exergonic reaction, which is essentially the irreversible process. Now the hexokinase, it is found in all the organism, and it has high affinity towards the glucose. Km value is going to be around 0.1 millimolar. Whenever we talk about the enzyme, the two values are very important for us. One is the Km value, which shows the affinity. More is the Km value, less is going to be affinity of that substrates towards the enzyme. Second value that we'll talk about is the kcat value, that suggests how quickly the substrate is going to be converted to the product. So these two ratio, if we take the kcat or km ratio, that will always give you the right value or the right kind of information about the enzyme. Now another enzyme that is into the picture, and this hexokinase can have other substrate as well, which are monosaccharide and hexoses, but for them they have the low affinity or the low specificity. Okay, and certain I mean this is going to be inhibited. This is going to be the allosteric enzyme by the product, that's the glucose 6-phosphate. Now glucokinase, it is found in the liver, and which has a low affinity. Look at, there'll be difference of around 100 times, where it is having the 10 millimolar km value for the glucose, and it will not be inhibited by glucose 6-phosphate, hence cannot be controlled. So most effective when the glucose level in the blood is relatively very high. Right after the meal, this enzyme will come into the picture, and they will take the glucose into the liver. The second step is the isomerization of the glucose 6-phosphate into the fructose 6-phosphate. We have the glucoisomerase or phosphoglucoisomerase. Here, the basic change which is going to take place is that the aldose or the aldehydic group will be converted into the ketonic group. So this is kind of a functional isomerism. Now the third step where the enzyme phosphofructokinase will come into the picture. One more phosphate group is going to be added. This is the second ATP investment. So fructose 6-phosphate will be converted into the fructose 1,6-bisphosphate. Here again the energy, and this process is essentially the irreversible process. Let me remind you, wherever we see the irreversible reactions, those steps are very important for the regulation purpose. So this is the one which is going to be modulating the carbon flux through the glycolysis in response to the energy and the carbon requirement. Just tell you the basic fact is that when the energy is sufficient, so glycolysis is not required. So that can be in the form of the breakdown product or ATP level that can matter. The fourth, the cleavage of the two triose-phosphate with the help of enzyme called aldolases. You can see over here. This is going to be broken into the fructose 1,6-bisphosphate is going to be broken into the glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate, that is a DHAP. So here the 3-carbon compound can be formed out of the 6-carbon sugar, and the delta G value is going to be positive. So driven by the one of the following reactions, and we have the triose phosphate isomerases which can keep on converting the DHAP into the glyceraldehyde 3-phosphate. Now, so this enzyme is required for the interconversion of the product, whereas the glyceraldehyde 3-phosphate required in good quantity, because this is one which is going to be utilized further in this reaction. Okay, here this process is going to be reversible, and this is a very important point which is required for the gluconeogenesis. If the glucose can be formed other than the glycogen, this is called as gluconeogenesis. For example, it can be from the lipid or it could be from the amino acid. So into the first phase, where the production of the 2-glyceraldehyde 3-phosphate from a single glucose molecule is going to be formed, and here we have expanded the 2 ATPs. So the product and energy yield of the following steps are multiplied by the 2 relative to the glucose. Now the second phase, that is energy payoff, now it will give you the energy. So oxidation of the glyceraldehyde 3-phosphate, you can see over here by the dehydrogenases, that will give you the NAD is going to be converted to the NAD. Roughly NADH will give you the 2.5 to 3 ATP per molecule. And first high energy compound generated beginning of the payoff product is the acyl phosphate, a fused carboxylic phosphoric acid anhydride, and which is going to be formed. And this reaction have a value around 6.3 kg per mole because of the fused group retains some energy produced by the oxidation of the aldehyde to the carboxylic acid. Look at over here. So the reaction produces the important reducing compound, that is NADH, and here the one the the compound is going to be the converted to the NADH is the NAD plus, which is a coenzyme, and this is the mechanism which is going to be involved. This will not discuss in very detail. So seventh step is going to be the transfer of the phosphate to make the ATP enzyme, phosphoglycerate kinase. You can look at over here. So here the phosphate group is going to be added over here. So 1,3-PG and 3-phosphoglycerate is going to be there. So it is the first substrate level phosphorylation. There will be two ways the ATP can be generated, one through the electron transport chain and second during the reaction itself. So that is called a substrate level phosphorylation. So the 2,1,3 bis PG will yield the 2 ATPs. Now the phosphate group is now be exchanging the part, like it is from the third carbon to it will go to the carbon number 2. Here also the energy is going to be delta G value is going to be 4.6 kg kilojoule per mole. Then we have the another enzyme enolase, which is going to form a double bond over here. So phosphoglycerate, 2-phosphoglycerate will be converted into the phosphoenol pyruvate, and enol phosphate is a high energy bond. And the final generation of the ATP enzyme, that is a pyruvate kinase. Again, the another ATP, second substrate level ATP is going to be formed during this process. So the phosphoenol pyruvate will be converted to the pyruvate. Now that I told you in the beginning, pyruvate will be having now the 3 part to follow. So this drives the several previous reactions. Pyruvate is a primary product of the glycolysis, and pyruvate is a central branch point in the metabolism. I told you there will be 3 ways, it is going to be used further. Now the bookkeeping, 2 ATPs formed from the each glyceraldehyde 3-phosphate. So total 4 per original glucose in the second phase. Out of that, let me first talk about the 2 molecules of the NADH also produced, and in the beginning we have invested the 2 ATPs that we are going to minus. So net gain is going to be 2 ATP and the 2 NADH. So summary of the energy relationship for the glycolysis: input is going to be 2 ATP, output is going to be the 4 ATP plus 2 NADH. So glucose to begin with, and the fructose finally will be converted to the fructose 1,6 bisphosphate, and this is how the reaction is going to happen. So energy yield from the glucose here is going to be around 2840 kilojoule per mole, but here the 2 ATP will produce the 61 kilojoule. That means at the substrate level phosphorylation, it is just the 2% of the total energy which is produced during the entire process, during the aerobic respiration process, where the Krebs cycle and electron transport chain both are operating. So as I told you, the pyruvate will have the 3 ways to go: alcohol fermentation, lactic acid fermentation, and aerobic glycolysis. That is how it is going to be. Aerobically, like pyruvate will enter into the mitochondria, where it will be broken into the CO2 and water. This is the part of the citric acid cycle or the TCA cycle, and that will yield more ATPs. Around 36 to 38 ATPs is going to be formed. Anaerobically, lactic acid fermentation, which occurs in the muscles and some of the microorganisms. And commonly, the ethanol fermentation is very common in the prokaryotic system or some of the eukaryotic system as well. For example, the yeast. Lactate fermentation, you can see over here, the pyruvate will, this is the process which is very important, in the 2 ways. One, the pyruvate will not have a fat to get end up with, so it will be converted to the lactate. And second, that is more important, here the NADH which has been locked over here, so that will be released into the NAD plus. So NADH is going to be produced or used, produced during the process, can be reused again. So this helps to drive the glycolysis by using the NADH, making the NAD plus available, reversible. So pyruvate can be regenerated in the alternative metabolism, and this is very, very important at the many places. For example, the RBCs, our retina, or the skeletal muscles, or when we do the strenuous exercise where the anaerobic respiration occurs and the lactic acid can lead to the fatigue into the muscles. And some of the plants and the microorganism, they also do the same thing in the absence of the oxygen. Now, the lactic acid which is generated into the muscle will have a path. You can see over here the pyruvate will be converted to lactate in the muscles, and this lactate will go back to the liver, where it will again convert it into the glucose, and the entire cycle we call the lactic acid cycle or the Cori cycle on the name of the discoverer. So liver release some of the glucose, and the liver uses most of this lactate to make the glycogen, and the glycogen can be broken down into the glucose when needed basically in the presence of the glucagon, one of the hormones produced by the pancreas, alpha cell of the pancreatic gland. Alcoholic fermentation, here the pyruvate will be converted first into the acetaldehyde, and you can know the once the further NADH is going to be used over here, and ethanol is going to be produced. So first step is going to be reversible, and second step is going to be reversible, and again the NADH which has been locked over here will be set free. So this pathway is active in the yeast, and second step helps the drive glycolysis, and second step is going to be reversible. In human, have the alcohol dehydrogenase in the liver, so it is mainly used for the disposal of the ethanol. Now come to the regulation part of the glycolysis. So we have the 3 irreversible kinases. So always keep in the mind in the metabolic pathways, wherever you have the irreversible reaction, those are always going to be target for the regulation. So 3 enzymes we'll see over here: hexokinases, then we have the phosphofructokinases and the pyruvate kinase. These are going to be used in the regulation part. First is the hexokinase, and the reaction it is causing the phosphorylation, and it is a kind of allosteric enzyme where the end product is going to inhibit the reaction. Second is a phosphofructokinase, which is a regulatory enzyme, which is a rate limiting enzyme. And here the inhibitor is going to be ATP, again the allosteric one, by decreasing the fructose 6-phosphate in binding on the citrate, because the ATP citrate will be available only when the energy is enough with the system. And if the energy is not there, there will be more ADP and AMP into the system. So that is going to be triggering the enzyme. And another way of the regulation, we have the one activator. The fructose 2,6-bisphosphate is a very important regulator, which control the relative flux of the carbon through the glycolysis versus gluconeogenesis. It also couples these pathways to hormonal regulation as well. Then the pyruvate kinase, which is formed because of the PEP is going to reacting with the ADP, and the pyruvate and ATP is going to be formed. This is the allosteric tetramer. So inhibitor is going to be ATP. Another inhibitor is going to be acetyl coenzyme and the other fatty acid. And activator is always going to be fructose 1,6-bisphosphate. This is the feed forward and this is the feedback inhibition. Then the phosphorylation, which is in active form, and the dephosphorylation is the active form under the hormonal control. It could be vice versa as well. So some enzymes are many ways they can be regulated. One, they require some proteolytic cleavage so that the zymogen form can be converted into the active form. They require the phosphorylation and dephosphorylation, and they also triggered by the gene expression pattern as well. So hopefully you have liked the topic. Do watch it and share and subscribe my channel. I'll be getting more in the future as well. Thanks.